You're listening to Barbell Logic,
the podcast where we talk about what it means to experience strength
and how you can use simple, hard, and effective strategies in training and nutrition to improve your life.
It starts with meeting you where you are right now and finding lasting solutions.
Welcome to the show.
Ahoy, everyone. Nikki's not here. I'm crashing the podcast today. CJ Gotcher with our good friend and staff coach here with Block, Jayson Ball. How you doing?
Hey, I'm doing well, CJ. How about you?
I am living the dream. Living the dad dream right now. Now, for our long-term viewers, this is not your first show on your first show with us. You were with us back episode 264. It feels like 50 years ago because it was January 2020. So,
it's crazy. That's been over 2 years.
So that's kind of where I wanted to start a little bit. So you are with the University of Colorado Boulder. You're not just a coach with us. You're also doing research in pain neuroscience. And that's a lot of what that previous podcast was about. You were talking about everything that was going on in your lab. What's changed since then? Like kind of cover that 2 years to today, where you're at, and then we'll go over all the exciting stuff.
Okay, so we'll go just real quickly with where I'm at. I'm in the middle of a PhD program. And the program is like a master's/PhD. Basically, it's just you're being trained to be a scientist. And the program is, in the beginning, you have a couple years of classes, and in the end, you have a few years of research. And in the middle, you've got this kind of like a titration from mostly classes to mostly research. And I've fully gone to the mostly research phase. So now it's like full-time lab, aside from a couple other obligations to fulfill the criteria for the PhD. So doing a lot of research, getting a lot of skills. We've got a little bit of a bigger team, and doing that, feeling like as the program goes on, I just have more times doing more reps. My form's getting better. Nice. in the lab world too.
The linear progression of science. Well, and so I'd like to actually talk about the reason, or at least the spark that drove this podcast was you published, or you were a co-author on a paper that published in the journal Pain.
Yeah, third author. And we can talk about what the authorship ranking or order means too. But yeah. Yeah,
absolutely. So you were third author in a paper that published in Pain back in January. That paper, if anyone wants to go look it up, preconditioning by voluntary wheel running attenuates later neuropathic pain via NRF2 antioxidant signaling in rats. Now, for us, this was kind of like fun to nerd out. And I remember during last year's block party, you were just to the moon, with everything that was going on, like talking, kind of talking, because you were deep in the research then. So a part of it that I was really curious about as a coach myself, and we end up reading the research and trying to stay on with it, is a little bit how that process worked. So you were about to wrap up, it sounded like, last August, and then it took till January to publish. Like, how did that train go? Well,
the final experiments, it was the first, second, and senior authors on the paper were doing, they were wrapping up some protein. There was a couple of experiments that were yet to go, but I had basically nothing to do with it at that point. And then the senior author, Suzanne, and the the first author and the senior authors. The first author was Suzanne. The senior authors were the two people at the end, Peter and Linda, did the main draft, and all I had to do was help with edits after that. So I was pretty much coasting on that particular project.
Okay.
While other people did Western blots and made the reviewers happy.
Nice. Now, so there's two threads I'm hearing from that then. One, I'd like to come back to the third author piece here in a second, because we always kind of assume it's like, there's 10 names on the paper. It's Morton et al., that paper that I reviewed. It's like, oh, Morton's responsible for this. Like, wait a minute, there's a lot of people in this project who have different roles. And I'd love to get with that. You also said there, well, I was on cruise with that project. And when we talked before this, you're now that paper was pretty recently ago. I mean, okay, 5 months. You're already moved on. And I'd say it sounds like you had already moved on by the time the paper published. So how is your research output looking like? Like, what is that? It's not like you're just doing one project and then, okay, that's done, next project.
No, definitely not. It's I think the best way to do research is to try to do as much as you can possibly handle. And some things will work and that you will have an interesting story. You will find that the technical things that need to happen are possible and worth it. And then some things you will not have an interesting story, or you will technically fail. And you will still learn things along the way. And that's kind of the phase of life that as a student you should definitely be in. And I don't know if you should ever leave. I'm sure that there are people who have like there are definitely more senior people who have like their own like expertise, and they know with like, hey, if there's a 90% chance that we can technically execute this thing and it will answer the question we have. But for me as a scientist in training, I think it's much better that I'm trying to do a bunch of different things and then focusing on the things that are sane after the fact.
Okay.
Because I don't know any better.
In that previous podcast, you were talking about 3 different channels that y'all were following: the toll-like receptor 4 inhibitors, the exercise, the recombinant gene therapy. And then this paper was along the exercise route, still with rats, and we'll talk about that here in a second. So it feels like if you put 5 pots in the fire, maybe 1 will or 2 will pan out. And
yeah,
so
totally.
You've got to keep your head in. So I've got to ask, when you mentioned that, like, do all the things you can as much as you can, I'm like, life balance, work-life balance. How does that work as a coach?
Well, I don't know if you know this, but I have like, I think I'm down to 9 clients, and it's it's all on block. And then we'll meet up like in person. Most of my clients on block are my pre-COVID in-person clients. And we transferred into block and they all bought home gyms when COVID hit, and they're still on block. And then now that they all have home gyms, we just do that. And we meet in person at a local gym where I just rent space by the hour. I don't run my own gym. I did that with Noah for a few years, and that would be impossible to do right now. But right now it's just like we do in-person coaching as a social activity, more or less. Like, it's good to get like eyes in person and all that, but it's more fun just like so everyone can socialize a little bit and feel some camaraderie. And but most of the coaching is done like, 1 hour in the morning, and then on Saturdays I'll spend 3 or 4 hours doing programming, and that's it.
So for you, with your priority being, the research side of things, and you were going through that taper, you're more student here, more scientist here, like you ramped down your coaching to match, to
Correct.
kind of keep that balance.
And the I had to do that to stay sane, because I'm not a very organized person, like mentally, and so when I'm juggled too many things, I'm less efficient at them. And I was juggling a lot of things in research. So I was like, okay, so I just have to have more hours to attend to this whole suite of things. Some people are very, very good at, hearing something once and then it fits into a neat slot and it's perfectly crystallized into their mind. Whereas I kind of think in circles and patterns in order to clarify things for myself. So I have to have this level of inefficiency I think is innate to my personality. And it's just something I have to make up for. So it's like, I have to, at the end of the day or at the end of the week, go through and see, okay, what needs to happen next week, and take time to do that. Or things will spiral out of control.
Yeah, you got to know yourself. It's so funny that, a couple of weeks ago, me and Nikki were talking about VIP, and like so many people, they're like, oh, there's the elevator of coaching, or the escalator. Like, I got 5 clients, now I need 10, now I need 15. It's like, it's got to grow. Like, hold on, is coaching the thing? And for you, like doing research that is furthering the next piece of knowledge, one piece at a time on pain neuroscience, that's your thing. That's
my favorite thing. But I also love coaching.
Yeah.
So there's I really like research, and I'm also extremely extroverted, and coaching interpersonal stuff where I can have lifting and coaching is technically interesting to me, and I really like helping people to be like accountable to the part of themselves that wants them to succeed. I really like that, and that's way more appropriate for like a barbell coaching relationship than inside the neuroscience department.
Hard to have a conversation with rats.
Yeah, and it's and hard to have a conversation with rats. It's rats and rat brains. It's more like technical. It's like solving little puzzles. It's collaborative in a way that I really like. And then the coaching side is very different, like in terms of the social dimensions. And I really like having both. They keep me feeling refreshed. I think if I did the same amount of hours in one or the other, I would have burned out years ago. But because it's split between things that are so totally different, it's like, you know how dolphins sleep with like half their brains at once?
I've heard that, yeah.
So I feel like it's kind of like that. Like I get like burned out a little bit on like being in a lab and doing that. Started to get a little fatigued, need to have that part of my brain sleep. Then I can go to the gym and I've got all this energy again and doing something totally different. And then by like bouncing back and forth can kind of stay recharged in a way that I think is, I think it's optimal for myself at this point in my life.
I love that metaphor. I mean, okay, admittedly, dolphins are just pretty much cool about everything. Now, so let's go there. So let's go into where you were at and where you were spending so much energy into the publication of this paper, for which you were the third author. Sorry for
Yeah, I don't want to steal Suzanne and Mike and Peter and Linda's thunder on that.
Well, let's quickly summarize like what this paper was about.
Sure. So there was a model that was discovered or worked out before I came in the lab, wherein wheel running for a rat, it's a common model for exercise. You let the rat have access to a wheel. Rats innately enjoy running on the wheels. And the model was showing that not only is it something that where if you have an injury or a pain state, the exercise can help the healing of the injury itself and the allodynia, which is the intensification of pain. That had kind of already been modeled out. What was novel with what the lab did before I joined was they did a model where the rats were given access to the wheel, or they were a couch potato. And what they found was if you let them run prior to an injury, then they recover faster. And depending on, I guess it depends on the breed, but the basic model is you have less pain for less time. That's the general story. And and to be clear, it's like after the injury, they don't use the exercise as a like medicine to treat the injury. It's just that something is different about the physiology of the animal that has made it so that when they are exposed to this like a stressor, and the model we use is called a chronic constriction injury. It's kind of rough, so I'll describe it, but just viewer, listener, beware. So you expose the sciatic nerve, which is like the sensory. As far as we're mostly interested in the sensory component, which is a bundle of nerves from the lower lumbar nerve roots, and that goes and innervates the hind leg. And so you expose that, you like gently take it out, and then you wrap these ligatures around it. So you induce a state of like persistent mechanical constriction and inflammation. And the prior running improved the outcomes of that kind of like aggressive assault to the nerve itself, in terms of like the animal behavior. And so the natural follow-up study is like, if you're a molecular scientist, is okay, well, what is different about the tissue? What signaling molecules are changed? Are the cells phenotypically different? There's a lot of things that we could look into, and then the main story that emerged out of a lot of hard work, which again I was helping with, but I was like one of the guys in the ship with an oar, and that was it. But I definitely did see a lot of the thought process and kind of the trials and tribulations of figuring out where the hell this main effect is happening. The main story that emerged and became the publication was this, that basically antioxidant response, endogenous antioxidant response, is primed, it's conditioned in these animals that are constantly dealing with exercise, which requires the creation of reactive oxygen species and then their management. After an aggressive nerve injury with this ongoing inflammation, the animal is better able to resolve the inflammatory or the reactive oxygen side of the damage to the nerve itself.
Okay.
So that was the paper there. There's all kinds of other mechanisms you could look into, but this one appeared to be quite key. It was actually like the ones that have the exercise. A good way of summarizing it is the same injury that hits them. It doesn't produce as bad of a phenotype, as painful of a phenotype, as much tissue damage, and they repair it faster. They're able to maintain like the massive amount of oxidative stress that's going to occur, free radicals, and prevent it from ballooning out into something larger, and thus reduce the total amount of oxidative stress damage on the tissue. And the thing that was interesting was that the main story Suzanne found was in the nerve site. So the actual site of damage. What she went into this thinking, and Peter went into this thinking, and Linda went into this thinking, and I tagged along naively, also thinking, was we would find more differences in like the central nervous system, where the signaling in the descending pain modulatory pathways would have conditioned the spinal cord to have less allodynia, like amplify the signals less, or the central nervous system, because we know after these kinds of peripheral nerve injuries, there's central sensitization, which is a phenomenon where the sensory system that basically that you can think of it, it's an oversimplification, but you can think of it as a relay. It's from like your fingertip to your spinal cord to your brain. That system, the immune cells around the neurons of that system become activated and inflamed and change the behavior of those cells. So our thinking was, the thinking at the time was, well, if there's this redox homeostasis, reduction, oxidation, homeostasis training, because they're constantly doing cardio, then maybe that's helping with the oxidative stress component of inflammation and that would reduce the amount of positive reinforcement in the loop of inflammation that becomes pathological and makes the animal persistently sensitive to pain and touch for months afterward. That was the thinking. It's like, we have this kind of like cute story. Wouldn't it be great if that was the case? That wasn't the case.
And instead, you found that it was actually the normal reactive oxygen species, the normal inflammatory response, the damage actually at the site of the injury that you see after you've surgically injured the rats in this particular instance. At the scene, the body was better able to respond to and calm down that injury instead of central sensitization.
That's right.
Instead of the nervous system.
That's right. And the thing that was interesting about it is when it wasn't just correlative. It wasn't just like, because there's some cool correlations. And then with basically, hey, we see more of this master antioxidant transcription factor. So for people who maybe haven't taken biology since high school, just a quick reminder, the transcription factor is a protein that looks for a location on your DNA, and once it binds along with its friends, the gene that's downstream of that transcription factor is turned on. So it transcribes the RNA for that gene. So now that gene is being expressed. And we found, look, the transcription factor, and actually I think Peter had found this while doing an RNA-seq, like a population level survey of the all the RNA in the tissue. But the basic story was like, look, NRF2, which is a transcription factor that makes antioxidants, like the body's natural antioxidants, was massively up with the wheel running. And what we found is, in this tissue, even weeks and weeks and weeks later, there's more of that system driving. Like the basic, like as a gym metaphor, it's like the animals have been getting jacked and in their ability to deal with inflammation. They've been doing reps of this transcription factor loop. And so when push came to shove and they needed to use that system, it was stronger. It was like, you've been getting jacked in the gym, somebody comes over and needs help with the couch. It's not as big of a problem for you as if you had been a couch potato.
Okay.
It's a stretch of a metaphor to apply to molecules, but that's the idea with that system. The system had been trained by the persistent stress of having to constantly rediscover homeostasis or reestablish homeostasis after running all the time. So that's, go ahead.
Well, that was something that really interested me when I saw your paper and when we talked about it, because there's sort of, it's a neat story, like you said, and there's sort of this coaching, like, yes, haha, this is one more piece of evidence, in the pile that, exercise is good for you. Exercise resolves pain, do exercise. And then, listening to you actually get into the details, it was also cool from a coaching perspective to be like, the different challenges and expectations that weren't met, or the first question that I had is kind of a lay reader, someone who's not as familiar with the research, is okay, kind of the injury that you surgically induced in these rats. There's a neuropathic pain, like directly to the nerve, and how similar is that as a model to what a lifter thinks about DOMs or maybe their own like surgical injuries, something like that.
So very different from DOMs. So first of all, like neuropathy is like defined as pathology to the neuron, and that is very different than like a temporary change in the sensitivity of that neuron that will reverse after a day or two. So they would require either like plasticity changes that are pathological in the context of the circuit, but more properly like actual dysfunction and dysregulation of the neuron's internal state. And so you can think of that as being like, well, we have a bunch of neurons. That's the nerve is composed of, is the axons of the neurons, and we strangled them. So it's real physical damage. This would be like the like the equivalent of like a very bad, I don't even want to call it like sciatica. It would be like a very pathological like bone constriction, like if you had a constriction that lasted a very long time. It was persistently pushing on the nerves and doing damage to them, like physical damage is what this would have to be simulating. Maybe like more like, maybe something like a car crash that didn't fully sever the nerve, but left it intact. Although after having a massive amount of physical damage, but not fully severed. There are other models that people use, and this wheel running model would be just as interesting to try with other models. And people do exercise, on how, neuropathy doesn't have to be just like, oh, we saw the nerve and cut it, or we constricted it, or we did a thing to it to physically damage it. You can do neuropathy by like inducing diabetes. It's like diabetic neuropathy, and you'll have nerve pathology that does chronic pain. And a good next question for somebody who does like a diapathic neuropathy model would be, hey, let's use the same exercise paradigm that they published and see if that helps prevent the pain we see with diabetic neuropathy. That's a great question, and because in our model it's like, well, it helps with dealing with this injury. Does that, and diabetic neuropathy is not the same kind of like acute injury, so maybe that's interesting to somebody to see if you get the same effect, and is it also oxidative stress, or is it some other mechanism by which exercise would be protective against like chronic pain?
Well, I think that's so. It's super interesting to me because there's a Twitter account called Just Say in Mice, I think is what it's called, where the number of headlines which like report just over the top findings from a study where the study was done in mice or were done in
Sure.
rats, and the headline never mentions implications like that. They never make the connection that either this was done in mice, people, calm down, we need to make that next step, or
Sure.
what this means in mice. Because to say, oh, it was better at scavenging reactive oxygen species after an injury means so many different things.
Yes. Yeah, that's a problem with journalism. Like maybe some scientists could like overhype their own the relevance of their own models. Like you see a little bit of that, not much, honestly.
Well, everyone.
I mean,
hopes-
you have to make the case of relevance to humans somehow. But I think that they like, hey, this whole thing is you have to make. I think for something like, especially something like this, like I don't know exactly what headlines you're talking about this Twitter account, and maybe you could post them up. But this is something what we call internally basic science. So the goal is not necessarily to be like, hey, look, here's another reason you should exercise, because we would not get funded if our goal was to find another paper that people could wave at their friends or doctors could wave at their patients to change human behavior.
Because that's worked for the last, 5,000.
Yes, exactly. It's like, it is nice to have, and you might as well add it to the pile. But the real thing, the real purpose is, hey, look, if we figure out the molecules behind this phenomenon, then that could open the door to using these pathways as a treatment. Or, if you had some sort of understanding of like a high risk situation where disease is likely or injury is likely, a preventative, that's a stretch. But it's like to understand the molecules of how this stuff works.
Sure.
And not to say, I guess like full stop, actually. I'll just say that. Full stop.
Yeah.
It's just to understand the molecular mechanisms involved, but because once you understand that basic science, you can have you now have something that in conjunction with other evidence, if there's a lot of evidence pointing towards some, say like a master transcription factor as being relevant and helpful for certain kinds of pathologies that have a hardcore dysregulation of oxidative stress homeostasis, you might have somebody who's crazy enough with their money to bet 5 million that they can push some solution through FDA trials that might work for helping some condition. That really is the primary reason. And then also, you get there's the romance of adding a leaf to the tree of knowledge. And then the university does have an interest in training scientists. So it's like, if you have, this thing where you have to be technically skilled enough to execute science like this, then you're making good scientists by doing that. And I would argue, being in the lab, for sure you'll be a better neuroscientist than if you just take neuroscience classes. But I think if you're in the lab and you're curious and good with Google and PubMed, then the neuroscience classes are like the filling in the gaps, but the bulk of the education is just experience. That's going away from your initial point about the, science journalism and headlines of, hey, they solved this disease in rats, therefore this disease is solved. Let's just tell everybody this disease is solved or something like that. That's
it's not even
annoying.
It's not even just on the journalist side, but also I think it's great for coaches too, because there are a number of personalities everywhere along the chain of knowledge. There's an urge, I guess, to what was it when Dr. Oz went in front of Congress, and they asked him, like, why did you advocate for all these things? He's like, well, I sell hope. I sell people hope. And it's that if anything, I think you made an excellent case for, hold on, yes, this is hope, but you don't know where it's going to go yet. This is basic science. These are the first steps. These are like, you don't know if learning this molecular pathway 20 years down the line will create a drug or will create something. We just don't know that yet.
Sure. Or it'll give somebody a target to say, hey, I had this population of people who are presenting with this cluster of phenomenon, or this cluster of disease phenomenon, maybe the gene that these people found was relevant to dealing with the underlying thing of this disease. Like, let's say you have a disease that's based on like the inability to deal with antioxidants. Somebody might pick up this and a bunch of other papers and say like, hey, I wonder if one of the proteins here, I wonder if the gene's messed up. And then look and do like, some genetic scans and then build it's not like it builds something that's not giving somebody a drug for the thing that is upregulated in exercise, but could be a genetic tool like, I don't know, a CRISPR or something like that can do a repair in the genetic code of the cells that are mutated or something like that. Like, this could go a lot of different ways by putting the knowledge out, the knowledge out there. And it would require, either a lot of audacity and willingness to make a big bet, or multiple lines of converging evidence from different model systems, ideally in different species, kind of converging on similar mechanisms. It's like, because that's when you have a big lever to pull, and so that's when people are willing to bet.
Yeah. Well, that's actually one thing I wanted to take it to. You were talking about multiple lines of converging evidence. So one of the first things that strikes me is, of course, it's like, this is cardio. What the rats are doing. And I have nothing against cardio. I love cardio. But I've always looked at and seen the research on analgesia and response from resistance training. And this is just from my lay background. I know you're a lot deeper in this. So this did not, this did not evaluate the effect of resistance training on the ability of the rats to effectively scavenge reactive oxygen
we
species.
did. I think Suzanne did look at she was the first author. Suzanne's the first author. She did look at the correlation between like the amount of exercise and the proteins of interest we were looking at to see if there's a dose response, but there wasn't one. It was basically they all like to exercise, so it was like they had if you had perfect compliance, then you got gains.
If only people were like rats.
Yeah, for sure. Yeah, this is. I think that the word from Linda was that the NIH is hesitant to fund exercise studies if the goal is like to make a direct connection to a human intervention with exercise, because they've said like, hey, people will not exercise like rats. Rats love it, and that's their favorite thing. And humans, some small percentage innately love it. Some learn to love it, and then even then, it's not something they want to do all day.
That is so interesting.
So it's like if you're going to go and fish for relevant molecular mechanisms, that you have a better shot of getting funding than if you just try to make it as a direct translatable intervention. It's not about hey, people should run this much because we saw this much in a rat and we think that dose would correlate to this much in a human. No. It's like, here are the molecular mechanisms involved. Those might be useful. That's the angle. But yeah, the rats, we didn't test their gains, so we didn't do like a pre and post first 1 rep max or anything like that. No strength gains, no VO2 max on that. But people do that. I saw a guy. I think that the wheel running thing is like an easy model because you get a really strong exercise effect because they love them. They love wheel running. I don't know if this is true, but I've heard that if you just like the researchers just put wheels in fields and monitored like wild mice, and apparently they liked running in the wheels a lot, and would actually like their behavior for like seeking food and mates would be reduced because they would like to play in the wheels. I don't know if that's true or that's just a myth that got propagated around the lab, but the idea is amusing. It's just like I just see like the wheel in the middle of the field just like spinning because little guys are in there.
I know it's so much easier, because years ago I saw, and I had to look it back up. It was too good not to miss. There was a paper where they're trying to develop a weightlifting exercise model to study hypertrophy in rats. And
which one do they do? What do they do?
Ended up coming up. It's a vest attached to a wooden arm on a fulcrum with weight on the wooden arm, and they've got a stimulator on the rat's tail that causes their leg to extend. And
so they did like an involuntary leg press?
Yeah, basically, like a standing
Oh, God.
leg press attached to a fulcrum arm. I'm like,
That sounds like a nightmare.
right?
Yeah.
So I can totally understand why going for this model, how it when you're looking for the molecular mechanisms, it's so much easier to pick something the rats are actually going to do.
Yeah, you just give them a wheel and then come back in 6 weeks. That's
Nice.
it. You don't have to train them. You don't have to manage them. You like make sure the cages are changed and stuff like that. But there are people who do resistance training models that I've seen. I haven't seen the one with the, what was it, electrically induced leg press model?
Basically.
Jesus.
It was essentially a weighted vest, a vest attached to an arm with a weight on it that they would stimulate, like force the hind limb to extend.
So I've seen ones that sound like less, well, they're not forced, ones that are not forced or take up all your time as an experimenter. I guess they still take up the same amount of time, but it seems like way less management. Basically people that have like I forget if it's like, they use like a treat or a zone that's like dark and the rat likes, like a little box, a little cubby, and it has to climb a ladder to get there. And then they just add weights. They do a linear progression. It's either a vest or they like wrap it around like where the tail meets the body. And they'll do multiple rounds of this, like 8 sets with a minute rest in between. And then they measure I think they did. I'm pretty sure they did grip strength and like size of their quads and stuff like that. After doing and they would test a 1 rep max, like keep adding 30 grams or whatever it is. I actually think it was 30 grams every rep until the rat is no longer motivated to go up the thing.
Here we go, folks. That's your. I mean, if you got your hamster waiting at home, here's how to get hamster jacked.
Yeah. So that was, so people have been doing that, but I think that the wheel running is just more popular or is more established in the literature because it's such an easy model to use. But if you want to get into strength training specific gains rather than just saying, oh, exercise versus couch potato, then you'd have to do a model like this, or you could use humans, but it's like running like really tightly controlled studies on humans is tough and time consuming and expensive, and you can't, and there's different ethics, obviously. So you can take
so difficult.
Yes. Well, yeah. If you're not a complete psychopath funded by other psychopaths, it's hard to do rigorous molecular biology on humans because you're basically limited to like, I don't know, saliva and blood and urine. Yeah, you can't just kill them at will, so which is good.
Yeah, that's nice. So actually, that's a funny segue into something I was thinking, and we had talked about a little bit before.
Sure.
But it's that the added layer of complexity, not just biologically, because obviously, we have the biology of rats is different than humans. Does it translate? But we also have the added complexity of our psychosocial environment and how we exercise. And when that comes into pain, things get real spicy.
Well, those are the main interventions for humans with pain are psycho and behavioral.
Yeah, and that's where I wanted.
There's some. I'm not as up to date on human stuff, but I know that there's some cases where things like very targeted sonic ablations work for people who basically have like a persistent constricted nerve problem, but the bulk of like just like a persistent allodynia, chronic pain situation interventions, where it's not some obvious, get the hatchet out of your leg situation or some analogous situation. Hey, you should, it hurts. Well, okay. Yeah. It's because your leg has a hatchet in it. So please remove it. If it's not something like around that obvious, and it appears to be more like central mediated, like not, if it's in the spinal cord and up, then the main interventions are psychological and behavioral.
Well, that seems, because from a coaching perspective, there's been a lot more talk in the coachverse, but also just in general about decatastrophize pain. So when
Sure.
someone has a tweak on the platform, when someone has that your first job is to decatastrophize, to contextualize, find out what they can do within the space, like, not to normalize the pain experience, but rather to normalize you're okay. You're going to continue functioning as a human being, and this
To
will
prevent
probably resolve.
catastrophizing.
Yeah.
Yeah.
And so like there's within the biopsychosocial, there's this feel like I can't remember who said it. They called him the psychosocialist, or he is an exaggeration of this mindset. He was critiquing the idea that, anytime a biological mediator is proposed for a back tweak, something that happens from training, then it's almost like rejected or ignored because this is the cause of pain. Or it's on the psychosocial side, this is the place that we can address. And yet I see you working in the lab in obviously a very different context with rats, but having effectiveness. And there's other research on pain specifically where there's clearly a biological, marker for it. So I've always kind of wondered from that, and we talked about it a little bit, like where does the bio come into play?
Well, for human interventions, you can't do a targeted, like the biological interventions, especially for like chronic pain, not just like, discomfort because you got a muscle tweak, but like chronic pain, like behavioral interventions, lifestyle factors, psychological factors, changing your relationship to pain. Those are the biggest levers to pull. Minimizing catastrophizing, which is like an actual, like pretty hardcore psychological construct. Like people, like it's like if you do like they validate it's a factor. It's derived from like massive factor analysis of pain patients, and if you know where somebody is on how strong they are in the distribution of total catastrophization, you can predict a large part of their recovery paradigm. So it's and their effects on their life in controlling for how bad the injury is. You could have like the same injury and the same subjective severity, and if the difference is the amount of catastrophization, like whether people are framing it cognitively as my life is over and there's nothing I can do, and this is it. This is all I'll ever be. Versus this is a temporary thing, and I will get through it, and that there's something they truly believe, then it has a large predictive effect on what their outcomes will be later. The pain human side of like doing actual medicine and talking or working with humans is something that's I've never done. Sure. But it's like I just come across it because like going to conferences, you see human stuff and talk, and you listen to human people giving talks on like human data, and I'm interested in the human literature. It's just not my strong suit. But my understanding in one sentence is most of the good interventions are psychosocial, So it's like if you in animals you can prevent, like you can cure. You can never really reverse the state of pain per se, but you can change it so that the allodynia no longer exists. You can we'll just say it. Yeah, you can cure the pain states in animal models. You can't take somebody who's got chronic pain and say, we're going to inject some viruses to silence some brain pathways. We're going to open up your skull and put a needle in it and inject some experimental viruses in there. But
that's a good thing.
Yes, and if you do something like cognitive behavioral therapy, then you're going to have a very low risk intervention, and that's the thing that has been where the most data is to say like that's where most of your leverage is, because if you have a pain population where you have a persistent chronic pain where you could actually say, let's say for the sake of argument, the injury has healed, but the feeling of pain is still here. Then it makes total sense to me that a very good thing to do for this person would try to get them to where they could live a life that is full of joy and not retreated in isolation and misery, because their body is telling them that there is a problem with the tissue, because that's what pain is evolved for, to like help you escape from dangerous shit that will kill you, and then to protect damaged things while they heal. But the pain system can, when it's pathological, is when the information it's sending, is the signal that it's sending is no longer related to that acute danger. And it makes sense that if you can do something to change somebody's relationship with their pain, that increases the ability for them to live a normal life that is not miserable, then that's what you do. And cognitive behavioral therapy is going to have other kinds of beneficial side effects as well. But I think what you're getting at with this is like, your question was, where is the place for the biological?
Yeah.
Well, so there's two things. Basically, like there's pain is going to involve, it's a product of the nervous system. The nervous system is a biological artifact.
Yeah, because it feels weird to say pain is only an experience. Pain is all psychosocial, because like our entire body to some degree evolved from somewhere, so it goes through. So it followed that process. It is a biological construct. The brain does not exist if the nervous system doesn't exist. So somewhere that has to be there.
Yeah, psychology does not exist if you don't have a nervous system.
Fair.
Yeah, so that's like the ontological argument. But again, like that's separate from what are the best options that are available in medicine in 2022. If that's what your question is, then it's like, okay, you have this suite of interventions that's largely psychosocial, behavioral, and then for like hardcore chronic pain, you could use morphine, but that fails and it has a lot of side effects. But just because the best interventions are psychosocial does not mean that the ontological like nature of the thing exists in a kind of ghostly like mist hovering above the brain that is only ascertained with construct analysis and psychology. It is a biological system, for sure. And you can change it in animal models. But because you can't do those interventions in humans responsibly or ethically, and it's not part of medicine yet, does not change the ontological nature of pain. It is based on the nervous system. Full stop.
And I think that's just like, I'm like, boom, he drops the mic. Because I love kind of where that thread goes, which is, yes, we as coaches, we have the place where we can intervene is in that general space that's going to work for most people. The decatastrophizing, the exercise, the preconditioning before the injury that builds the resilience, even if we don't quite understand all the mechanisms that go into it.
Yeah, you're helping people be healthy and active and the best version of themselves, like, and having a habit that is making them strong.
And then people like you and your senior authors and your first authors who are going through and toiling through the murky, misty side behind, beneath the water, so to speak, of the biological that we can't quite tell yet. Maybe 20 years from now, we'll have more from that.
Yeah, I think very soon, actually. I think you'll have some pretty solid, for certain kinds of pain states that involve chronic sensitization of the nervous system. You will have, for certain states. It's basically, I think the interventions are going to start most distal. So like something you have like knee arthritis. I know is one that's in human trials for for pain relief for knee arthritis, which there's no really good cure for. But there's gene therapies that are being tested right now, and in phase 3. We'll see if they get approved. But I think that the way I would, if I were to put money on it, I would say you'd have some reasonably good interventions for allodynia, and we'll just say at least like the allodynia portion of pain.
That sensitization, that making it
5-10 years. Yeah, that's right. Sorry. Yeah. So the hypersensitivity to pain is what we call like hyper nociception. Allodynia is the feeling of normal touch, is where normal touch is perceived as pain, or those wires get crossed, and the touch system is now driving the nociception system, and so and there's a system that's touch. It's touch, but it's also like crosses over and receives input from nociception. So if it gets set in the spinal cord, these are talking about like ascending sensory input to the brain. You have these strictly nociceptive pathways, more like strictly touch pathways, and then this area that's called like of where in the spinal cord they're called like wide dynamic range neurons, where if they get touch or nociception, they send signal to the brain. So after that neuron, after that synapse, the brain has just this input that is touch or pain. It's just like kind of this intensity of the stimulus. Either one's going to make it go. And allodynia is where something like after a sunburn, you put on a sweater, the sweater can hurt. So if you have chronic pain, that's the way everything feels. Somebody like your wife, your husband, boyfriend, girlfriend touches your skin and it feels like fire. That's a sensory pathology. I think that kind of stuff will be, if I had to put money on it, it will be, I think that will be largely a solved problem, within 10 years. 20 would be, 20 would, and at least in certain human applications, I think in 20 would be, it could be quite broad in 20, but you'll start to see at least some human disease states where sensory pathology of that kind, where touch is felt as extreme heat or extreme cold or as raw, like nociceptive. I need to escape from this thing because I just pierced my hand. Like touch felt like attack through the hand. That kind of sensory pathology, I think, will be you will get human treated where that is no longer their perception of their world in the next, I'd say 10 years. Yeah, I would definitely bet on that.
That's awesome, boss. Brave new world. Well, thanks so much. Like, honestly, I had a blast at the last block party talking about this, let alone, like us kind of pre-talking about it a little bit, and just generally discussing not just your paper, but the bigger piece to it. The one last question I have, because again, I was so, as you're like, rattling off all the things you're working on before, you were covering, like I said, toll-like receptor 4, exercise, recombinant gene therapy. What are you working on now? Okay,
so there's 2 projects that we're doing. The one I'm most excited about is actually following up on this really cool neuroimmune phenomenon in the spinal cord, and like this primary sensory allodynia that Peter did before he moved to Texas, where you basically have nerve pain, you have established chronic pain, and you treat it with morphine. There's this pathological effect. The mechanism is neuroinflammatory. But basically once the acute pain relief has weared off, you have residual inflammation in the central nervous system and a enhancement of allodynia. So it's a paradoxical, very sad effect of having treated with morphine after neuropathic pain already exists. And he figured out he and Mark Hutchinson and Suzanne and Linda and people came before me figured out all these immune mechanisms that were relevant. And now I'm working on a project with the current team to show, these immune mechanisms equal more pain. But where is this happening? And we're showing that the neurons of the pain pathway are more sensitive, and we're doing like hardcore molecular neurobiology, that they are more sensitive and excitable after this morphine treatment when pain is already there. So this morphine story is quite interesting. And then we're doing some like and that's a basic like sensory processing, and I really like that. That's my favorite stuff. And then I'm doing this other thing with this really excellent grad student who came here from Brazil. And actually, he's going to get his PhD. He'll be done in 2 weeks. I think 3 weeks from today. He's going back to Brazil to defend, and he's coming back to keep working with us.
Nice.
And anyways, so we're playing with this. This is more like basic neuroscience of just like how does the brain work stuff, and we're playing around with very fun neuroscience tools to delete a pathway in the brain, so that like instead of having to like basically just blow up a lesion, a region of the brain to see, oh, what does this thing do? Well, let's just destroy it and see what's different. And that was the way people had to do science before they had like these genetic tools. So now we can have like a nice logical intervention where it's like you can gently, say like, okay, if you're a cell and if you're a neuron that lives here and you talk to a neuron over here, gently commit suicide. Thank you. And you do that, and then you see the effects on these downstream pain processing things by deleting certain populations of neurons. So I'm really excited. There's this. I'm studying a pathway in the brain as basic neuroscience, and I'm studying the sensitization of pain projection neurons from spinal cord to brain. So like basic sensory pathway stuff after in like chronic pain plus neuroinflammatory state. Plus or minus neuroinflammatory state, and looking at the changes to the neurons there. And those are I'm excited about both of those projects. And I think for a PhD you're supposed to just have one, but I actually can't. I don't want to drop either of them. So I'm pushing both forward. And Linda, my boss, was really gracious and was like, "Hey, yeah, you should do both of these, and we'll just hire help." And so I've got like a really great team right now that we're all working on these projects and getting to do cool technical stuff.
That's awesome.
Yeah. So that's my world, and it's getting extremely fun.
All right. Well, that sounds like a great place to end it. Jayson, thanks so much for hopping on the podcast and talking nerdy with me. And,
up to talk nerdy anytime.
Got you around.
Cool. See you, man.